I'd like to get an idea of your background. Since we're biologists, we're microbiologists and biology based. Have you had biology in high school? Good. All right. Anybody had biology at the university level? Good. Anybody had a class in bioengineering or microbiology? You're a bioengineer. Chemical engineer. Okay. All right. So then we don't necessarily have to start from absolute basics. You probably know a lot of the basic terminology. That was one of the things, by the way, that the Clean Tech Open drilled into us, is that you really have to use common terminology. A lot of people that you see in this business, especially venture capitalists, really have very little background in biology and almost no background at all in microbiology. So what we have done at HelioBioSys is we have developed a process for producing sugars at a very, very low cost. And I think you realize that cost is really what is hampering the biofuels industry and the bioproducts industry today. So that's really what we have done. And let me just give you a little bit of history here about myself. I'm really a microbial ecologist, and I do a lot of field work, and I look at environmental systems and how they work together. Okay, and in addition to that, I've had a lot of experience running a lab. I've had graduate students, undergraduates, and so I've run the gambit there. David and I first got together many years ago on a project developing a bioreactor and designing a bioreactor for specific purposes for NASA. And although I'm going to stress in my talk that we don't do anything with genetically modified organisms, I'm not against using genetically modified organisms in certain circumstances. In fact, I was a founding member of NASA's synthetic biology program. So, David, if you can take over.
I've got a microphone here too. So good afternoon, everybody. And I got my PhD here at Stanford many years ago, and I don't recognize the campus anymore. So that's why we got a little lost coming over here. It's changed a lot, but at any rate, I focus on plant physiological ecology, looking at the interaction between organisms and the environment, and coupling biological and engineered systems. And I did that at NASA Ames when Rocco and I were there, looking at controlled ecological life support systems. I've also spent a lot of time doing ecological restoration here locally around in Palo Alto, community-based stewardship, using my ecology to help, protect our natural areas, which is, something that's a passion of mine. As Rocco mentioned, we've done a lot of design and development and operation of the bioreactor. I've worked with plant growth systems, and also we originally started with this mouse and algal system looking at gas exchange. So a lot of coupling biology and engineering together for long duration space missions, but for now in HelioBioSys, looking at industrial applications of how you put biology and technology and engineering and industrial processes together and make them all work. So looking at the mass and energy balance of those, and then I've also done quite a bit of government service around here locally. So I'm going to hand it back to Rocco to tell a little bit the story about what our company does.
And how it actually got founded in some of the original ideas. Well, way back in 2005, I had an idea that said, well, because biofuels were coming into play then, and everybody was really interested in biofuels and ethanol production. And one of the ideas I had was, let's just see if we can take the sun's energy and turn that into ethanol. Not quite directly, but almost. And the idea I had, which someone else came up with, at the same time, basically, was to use organisms called cyanobacteria. Okay, and that's what we still use. Cyanobacteria used to be called blue-green algae, but they're not algae, they are bacteria. And I said, okay, let's see if we can genetically modify a cyanobacterium to produce ethanol. And we did. We were successful. But there were many stumbling blocks. One of them was we got it to produce so efficiently that it killed itself. Second one was, okay, well, let's see if we can modify it and control it. Well, there's many things you can do with molecular biology, and we did it. And so we could control it. We actually could control the production of ethanol, and they said, okay, now we can do it, and it's not going to kill itself, but our productivity decreased. The next step was to see whether or not we could actually start doing it in larger than a test tube, go to a flask. And when we did that, we saw that we were losing a lot of our product through evaporation. So finally I said, this is just getting too much. And we're doing too many things we have to do. It's not going to be efficient, never. And I don't know if any of you follow the biofuels industry at all, but I said that there was someone else doing it essentially simultaneously. Well, they didn't give up. It's a company called Alganol, and what Alganol did is essentially took cyanobacteria and they put the same genes in it that we did, and they produced ethanol. However, I kept track of the company. By the way, they are still not doing well at all. They're not commercial, not on a commercial scale at all, because they had exactly the same problems we have. And what they've done to try to eliminate the evaporation is they now have little ponds, and they put a tent over it. And they have the ethanol condense onto this tent. And still, that seems to me not like I still think I made the right decision in abandoning that idea. They would disagree. So that's how it all began. And that's how we started with cyanobacteria. And for that project, we needed cyanobacteria that would grow very efficiently. But then, and we found them. So we had a bunch of isolates. Okay, come on. There we go. And we started off, believe it or not, at a place called the SETI Institute in Mountain View, California. Now, many of you probably have heard of the SETI Institute, and the way it got founded was it was the search for extraterrestrial intelligence. Well, there's another part of it that is astrobiology, and I do a lot of astrobiology. But this was an idea we had separate from astrobiology, separate from the search for extraterrestrial intelligence. So we got started at the SETI Institute, and that's where we did a lot of our development work, and that's how we essentially did a lot of the lab work to come up with our consortium, try to come up with our consortium, which is a group or a mixed community of cyanobacteria. And we specifically chose marine cyanobacteria rather than freshwater cyanobacteria. Most of biofuel companies use freshwater. We use marine. We use marine because marine organisms, especially cyanobacteria, normally and naturally make sugar, but unlike freshwater bacteria, they excrete it outside the cell. So you don't have to break the cell open, and you don't have to extract it. So we then started to characterize, put together our specific consortia, and we scaled. Okay, this is what a lot of people, VC people, they don't think very much of. We considered it. We scaled from test tubes to 150 liters in an open culture system outside. This 150-liter open culture system happened to be an aquarium. So we thought that was it is a big deal, actually. Let's see, here we go. Then we decided we had to figure out how we were actually going to go outside of a laboratory and actually get something done. And could we do it? We incorporated ourselves back in 2010, formed a company. We're a C Corp from Delaware. And so we decided we needed partners, and one of the partners that we needed and was willing to partner with us was SRI International in Menlo Park. And they have an excellent facility for conducting chemical and biological analyses. Then we said we need somebody who has some expertise in pond building and pond and cultivating organisms in ponds. So we reached out to other companies, Helia being one of them, who has that ability, because we're basically researchers. And when it comes to construction and engineering, that's not us. And we realize that, fortunately. And most recently, we also have formed more partnerships, because as a small company, you can't really do much yourself. So not only with SRI International, but we also partnered with Lawrence Berkeley National Labs, the ABPDU unit, and Moss Landing Marine Labs, which is part of the Cal State system, and a fish farming company called Monterey Blue Water Farms. They're an aquaculture company in Monterey. And we got together and we wrote a proposal to the DOE, because we are at a very early stage, even yet. even though we've been working on it for a long time. We're at a very early stage, and you can't really get venture capitalists to invest in you when you're this early. They want more of a sure thing. So the government hopefully will see their way clear this fiscal year to give us some funding. So the idea here is to do all of that and eventually build partnerships, because that's what it's going to take for commercialization. So our company really, we have to do all along the way chemical and biological analyses. And we have SRI International and Lawrence Berkeley National Labs to do that. We also have to scale up, from 150 liters to millions and millions of gallons, billions of gallons. But the first stage is to go to thousands of gallons, and that's what we're going to do with Moss Landing Marine Lab, and go to a 1-acre type facility with Monterey Blue Water Farms. And David will expand upon that later on. And then, of course, one of the biggest problems, that we have is harvesting that sugar. Okay, I said we have a marine system. It is. So that, and we don't have to extract it. That's good. But in a marine system, that means you have seawater. So what do we have? We have salty sugar water, even when we take the organisms out. Now, what's the next step? Well, you have to separate the salt from the sugars. And so and that's an engineering problem. So we're working with a couple of companies, Novacep and Dow being among them, to actually develop a system it's probably going to be based on microfiltration that'll separate our sugars. It's not, our particular sugar water solution is a little bit different, but this kind of technology of separating sugar and water, especially at a company called Novacep, they seem to do that regularly. So when we called them up and said, can you do this? They said, yes, we just have to design a system that's more fit for you. So it's almost off-the-shelf technology. And then, last but not least, when we do this, how much is it going to cost us to produce this stuff? And how much can we sell it for? And are we going to be competitive? And to be able to do that and figure out which part of each of the systems that we have costs and how we can tweak it, we need a techno-economic analysis. And in fact, we've started that already with the NREL has, and David's going to talk a little bit more about this, NREL has a special thing called NCAP that they will allow small companies 40 hours of time, and they'll develop a model for your system, and we have. David will cover that later. We're also, SRI is going to expand upon that, as well as Lawrence Berkeley National Labs. We've also developed somewhat of a partnership with something called Life Cycle Associates. So one of the questions that came up was a comparison of energy density. We don't have a pointer, do we? That's okay. All right, so this is one of the things that I just quickly found and put up here. And if you look at natural gas, it has an energy density of a little over 40 megajoules per kilogram. If you look at wood, that has under 10. And ethanol is a little above 40. And you look at crude oil, we're approaching 40. Maybe, I think it's 42. Oh, thank you. Now, figure out exactly how to use this. Oh. Now, if we look here, if you look at, we say we make sugars, and we do make sugars. So where would sugars lie on there? Sugars are about 17 to 20. Okay. That's not as good as natural gas or diesel or crude oil, but it's not bad either. If you look at proteins and other biomass, they tend to be a little bit higher, but not much higher. So we're all talking about basically within the same order of magnitude here. The other thing you have to consider here is that when you're talking about crude oil is, as you know, time, temperature, pressure, all of that stuff. Crude oil has a big advantage in that meaning the industry, has about 100 years or more worth of infrastructure to actually handle that. And to come in as a small upstart biofuel or whatever, you can get intimidated easily. Well, I wouldn't say intimidated, but it's a big behemoth. I do recall that one of our first presentations was at a company called Chevron. We went to world headquarters, which is right over here in Richmond. And when we sat down and started our presentation and talking about ethanol and ethanol possible ethanol production and our sugar. And really realized there that they use billions and billions and billions of gallons, even 10%. And one of the things that they said is, we don't necessarily think that you can make enough to make it worthwhile. Correct?
Right.
Because it just overwhelms you. One of the things they have there is you can spot their tankers as they come across the ocean. And they're just a line. You know, it's amazing. Well, it was to me. Maybe not to you since you're engineers, but it was amazing to me that you can just see the tankers are just lined up coming across the ocean. So you have to appreciate the volumes that you're trying to deal with here. Now, if we look at this energy density here, we have to make an awful lot of sugars to actually meet the crude oil. About, crude oil, I said about 17, and crude oil is actually, I think, about 42. Okay. Just to put things in perspective here. Now, cyanobacteria. These are our guys, and we put several of them together in a mixed community. They are a very old lineage. They are bacteria. They're not algae, as I said before. And in fact, 3 domains of life: bacteria, archaea, and eukarya. They're part of the bacteria. They are the first oxygen-producing organisms that evolved. They essentially gave us our oxygen atmosphere. They're found everywhere. You find them in lakes. You find freshwater lakes. Saltwater lakes, actually you can find cyanobacteria in salt crusts. You can find them in deserts. You can find them living inside sandstone. You can find them on top of rocks. When I say they live everywhere, they live everywhere. They are also hardy. That means, and they're well adapted to living in a variety of environments, obviously. And the big thing that we found and that we like is that the marine type organisms excrete the sugars outside the cell. The other thing about them, why we chose them to work with, is they happen to have the highest photosynthetic efficiency of all photosynthetic organisms. That means that they are very efficient at capturing photons and changing those photons into sugar or biomass or protein. Very efficient. Now, this may look a little busy, and it is, but I'm only going to use part of it. But what I want you to appreciate here is, if you look at the bottom here, you'll see that this is a timeline in billions of years. Okay? And this gives the Archean, the Proterozoic, and the Phanerozoic, what they call the different time periods. And this is actually taken out of last week's Science magazine. And so if we look here at the cyanobacteria, the origin of the cyanobacteria is thought to have occurred about 3.5 billion years ago. So they're a pretty ancient lineage. And they're an extraordinarily important lineage because they did give rise to the oxygen on the Earth. And here, if we look at the oxygen levels of the atmosphere as a percent of the present day atmospheric level, where 100% is where we are today. Okay? Originally, the Earth was anaerobic. It was 0. But then cyanobacteria evolved, and the cyanobacteria eventually evolved a very efficient mechanism for making energy, and that was photosynthesis. And the most energy you can get is actually using oxygen. So they developed oxygenic photosynthesis early on. Simultaneously, by the way, since oxygen is really very, very toxic, they had to evolve a mechanism to detoxify the byproducts. And so they did that. So it took them a while to evolve the whole system. And they lived in microhabitats or microenvironments. And so if you look at the rise, this line here is actually the rise of oxygen through time. And so the other thing, not only did they were making oxygen and getting a lot of energy out of it themselves, but when they made the oxygen, and it's so energetic that other organisms around started sniffing around and got the oxygen as well. And then they started to utilize oxygen because you can make so much energy out of it. And so eventually what happened, as the oxygen level rose on the Earth due to these cyanobacteria, you see the eventual evolution of eukaryotes. Those are the bigger cells that have a cell nucleus. And then it really starts to rise, and right up here with this, at this time period, you see that there's a lot of oxygen in the atmosphere. We're nearly at where we are today, and that simultaneously coincides with the evolution of, and what's in a real big burst in diversity of life on Earth. And there you got multicellularity, the rise of animals and plants, and everything that we see today. And it's all due to these guys here, the cyanobacteria. That's why I think they're really quite amazing and important. And how do you know, by the way, how does one actually try to figure out what the oxygen level is 3 billion years ago? Well, one of the ways, you can read the paper, it's in last week's Science, is they look at the isotopes of chromium, and they look at the oxidation state of the isotopes of chromium and other things, and you can see that it's very, very low, non-oxidized, and then it spikes and becomes much more oxidized. Now, cyanobacteria are also important because not only were they first photosynthesizers, but they are also the same organisms that cause photosynthesis in plants today. They're responsible solely for all oxygenic photosynthesis in some way or another because there's a process called endosymbiosis. And what happened was the eukaryotes, essentially, those bigger organisms, ate the little bacteria, but instead of just eating them completely and digesting them, eventually what happened is the photosynthetic apparatus in the cyanobacterium survived inside the cell, essentially became much more simplified, became part of the cell, and it gave rise to what we call chloroplasts today. So chloroplasts actually are ancient remnants of ancient cyanobacteria. Okay. And then the early first organisms here, eukaryotic organisms, eventually gave rise to the green algae, which gave rise to the land plants, also gave rise to the red algae and protists and other photosynthetic organisms. By the way, we talk about oxygenic photosynthesis, but because I am a microbiologist, I want to throw out there that there is anaerobic, non-oxygenic photosynthesis. They don't use water. That's the other thing I want to make sure everybody knows, as a general thing to know, is that oxygenic photosynthesis, the oxygen comes from water, not from carbon dioxide. So these organisms, by the way, they get their carbon from carbon dioxide, but it's not part of the photosynthetic system. Okay, so let's just look at cyanobacteria versus algae a little bit. Okay. They are prokaryotes. That means that they do not have a cell, a nucleus, membrane-bound nucleus, excuse me. They are bacteria. They are small. They can range from about 0.5 micron to 2 microns. Some of them can be filaments, and they grow in nice, long, slimy strings. They use photosynthesis to get their energy. They get their carbon from carbon dioxide in the air, and some of them actually get their nitrogen from nitrogen in the air. Bacteria, not a lot, but some bacteria actually don't need fertilization or anything. They can just get the nitrogen out of the air. It's a very energetically expensive process. They are green because of the chlorophyll. They live, as I said, essentially anywhere or everywhere you can find them. Not the same organism, but the cyanobacteria in general. They are vegetative and they go through binary fission. Now, if you look at algae, for example, algae are eukaryotic. They're important in the biofuels industry. They are not plants. A lot of people think they're plants. They're not. They're chlorophyta. They gave rise to the higher plants, but they're not. They are eukaryotic. They're not plants. They tend to be bigger. They're about 10 microns or more. They can be unicellular and they can be multicellular. They also get their energy from photosynthesis. They also fix CO2. That means they get their carbon from the air. But they don't fix nitrogen, so they have to be fertilized. They're green because they use chlorophyll for photosynthesis. They live in water, and they can reproduce either vegetatively or sexually. So, now if we look at our system and algae, especially centering upon the biofuels industry here, what you can see is that we use a mixed population of genetically modified organisms. Most algae that are used for biofuel use pure culture, and a lot of them are genetically modified to be much more efficient. We are counting on using some type of managed open seawater small acre pond minimally managed. They have tried, they, meaning, excuse me, the industry has tried to cultivate algae in open water systems. They use freshwater systems primarily, and they found that it's been very difficult. So a lot of the companies, such as Solazyme, has gone to bioreactors and non-photosynthetic systems, even though they are using algae. Algae require fertilization, and the feedstock, such as the lipid or fats or whatever they're trying to make, are usually inside the cell. So the process has to go through a dewatering process, and then they have to extract it. We don't have to do any of that. Products there are usually lipids or oils. We make sugars primarily, and the end product there is for fuels and nutraceuticals, and ours too are for fuels, pharmaceuticals, nutraceuticals, and bioplastics. And with that, I will turn it over to David.
Thanks, Rocco. So you can tell Rocco is real enthusiastic about cyanobacteria. And so when I first got involved in this, it's a very interesting group of organisms, and we really tried to pick features of that organism that would overcome some of the hurdles that we see in the biofuels industry. And one of those is the yield and the productivity, the land area that you use, things like that. And so there's a concept called biomimicry, and you've probably heard of that. It's like Leonardo da Vinci modeling his flying machine based on bats, right? The flying of bats. Or people have used biomimicry, the pads of tree frogs, to design better tread for tires, for example. So in biomimicry, you're taking a feature of an organism, an element of an organism, and you're using that to solve a complex human problem or to solve an industrial application. For example, Velcro comes directly out of the burrs and seeds of some certain kind of plants that people looked at that microscopically and then designed Velcro from that. So very useful concept. And so extending that a little further beyond an individual organism or an individual feature of one organism is the concept of ecomimicry. And that's a broader concept to look at, features of the natural world, ecosystems, or in our case, we're looking at groupings of organisms. We're putting together groups of cyanobacteria together so that they're more robust, they're more resilient as a community. Single organisms, just like a monoculture in farming, right, doesn't do as well as if you have multi-cropping. So our concept here is to put those organisms together, the nitrogen fixation. The ones that are good at fixing nitrogen are not the ones excreting the sugar, but they're internally getting this mass and energy flow going within this community of organisms that is increasing the productivity of that system, increasing the resilience of it to predation, contamination, things like that. So trying to use this concept both in terms of the organisms and the ecology of those organisms and how we can take that community structure and translate it to an industrial application, but also how do we use ecomimicry as a concept to build an integrated, tightly coupled mass and energy balanced industrial system that is actually producing biofuels, bioproducts, food, it's energy and water efficient, land area efficient. So trying to bring all those things together along with engineering, right? I mean, engineering is an important part of all this because it's not just the biology that we have to get right to make the economics work, we have to get the engineering right as well. So just wanted to introduce those concepts as something that is a company that we've been relying on. Now, just to sort of drive the technology home a little bit more, we're talking about open saltwater ponds. And you've seen, open algae ponds and things like that, something very similar to that. And our basic input material is sunlight, seawater, and air, right? So we have very low cost, reliable, abundant raw material inputs for our process, going into this consortium of cyanobacteria and getting sugars out of that. And these would be land-based ponds. This slide's a little bit we're not trying to do this in the open ocean. These would be land-based ponds, of course, near the ocean where you're not pumping seawater, and putting that together. Now, to look at a little more schematically here. So here's our inputs coming in, the group of organisms. That's an actual photomicrograph of some of the organisms in our consortium, and the products that are coming out of are these fermentable sugars. Now, these are monosaccharides, polysaccharides, so things like glucose, fructose, things like that, but also longer chain, larger molecular weight sugars that might be, directly convertible into bioplastics, or that you might break those longer chain sugars down into the fermentable sugars that things like yeast and E. coli would use to process. So that gives us this feedstock supply, and then that biomass conversion is how we're trying to convert these into useful products. So this artificially assembled consortium of cyanobacteria, we're projecting a yield of about 100 tons of sugar per acre per year, and I'll look at that a little bit more because that's sort of a key laboratory finding that allows us to compare ourselves to things like algae oil, to lignocellulosic sources, to corn, to cane, and things like that. Okay, so we've talked a little bit about how these sugars and what they go into. So this is just more of a schematic diagram of directly processing them, polysaccharides into polymers and things like that. And the goal is, if you imagine a barrel of oil, to be able to compete with oil, to be able to sequester and leave fossil fuels in the ground, which is the best way to combat climate change, right, is to not take it out of the ground in the first place. You've got to replace that barrel of oil. Well, barrel of oil is not just gasoline, right? Gasoline is kind of the waste product of taking all the good stuff out of a barrel of oil. And all the good stuff goes into pharmaceuticals, it goes into plastics, it goes into other, products. All the products that we use really are coming out of that barrel of oil. So there's a lot of people who are working on the classic fermentation into ethanol and butanol, but using these novel organisms, using genetically modified E. coli principally to make a specific molecule like adipic acid that then goes into nylon, things like that. So these sugars are the foundation upon which all of that being able to replace that barrel of oil comes into, and it's the price of those sugars and the variability in that price that really has a big impact on the profitability of biofuel and bioproduct market. So that's why we're really focused there. And as Rocco mentioned, Solazyme actually grows algae in the dark on sugars, and they produce cosmetics from some of that, and they also produce lipids for biofuels out of that. So a lot of different ways that these sugars can be used, and what we're trying to do is trying to build the foundation of that, pick a niche industry to scale this process up, and then have it expand into that broader spectrum, because you can't tackle all of those things at once. Now, just to give you a little bit of sense that we have done this in the laboratory and worked on that, and we talked about the photosynthetic conversion efficiency. TDC and TOC are total dissolved carbon or total organic carbon. So here's our consortium growing on raw seawater, which is how we do it in the open ponds. Here it is growing on a defined media in the laboratory. And here's a freshwater cyanobacteria growing by itself on a defined media. And you can see that we can produce a lot more total organic carbon than the freshwater things. And this is this inherent characteristic of the marine cyanobacteria to be able to do that. And you can also see the photosynthetic conversion efficiency. So this is photons, and this is the photosynthetically active photons between 400 and 700 nanometers, the ones that the photosynthetic apparatus picks up directly to sugars. So this is not just photosynthetic efficiency to biomass, but directly to the sugars. And you can see that we're significantly higher in our consortium, and that's really the foundation upon which we think that this can actually begin to, at least make a dent in some parts of this industry segment. And then on the bottom curve, the red curve here is actually showing the growth of this of our consortium with no added nitrate. You can see that actually is growing faster, better than some of the organisms growing separately or growing a different consortium on a defined medium with nitrate. So we have been able to do, some bootstrapping stuff in the laboratory and need to, get financing to really take it to the next level. Talked about the productivity just to make it more graphical. Corn and sugar cane are sort of the benchmarks now for bioethanol, and they're about 5 tons per acre per year. We're estimating 100 tons per acre per year, so 20 times more efficient is our target goal, and that's what we're aiming for in the DOE grant. If we get that going, that would be our target productivity. Another way to look at the productivity is to look directly at corn and sugarcane, and this is gallons of ethanol per acre per year. So corn, sugarcane, miscanthus is a grass crop that's sort of the cellulosic conversion, and that would be something where you'd take those cellulosic sugars, break them down, use those to produce ethanol. Now, it's not a direct comparison really, because we're sugars, algae is producing biofuels, but they do conversions to look at gallons of gasoline equivalent. So this is all on equivalent scale, and you can see we're about 8 times more efficient than miscanthus, which is something that's attracted a lot of venture capital money, a lot of grant funding and things like that. So we're really looking at trying to take advantage of the advantage of our system, and a lot of those competitive advantages that we're not competing for land, we're not competing for food, right? We're not competing, for land crop. These are, marginal lands. These are lands near the near the ocean. It could be desert lands near the ocean. So you're not competing with food. You're not competing for land. You're not using fresh water. I mean, we would use some fresh water, but nothing like modern agriculture. Nothing like the processing and growth of miscanthus. And nutrient resources. We're not using fertilizers. We're not using pesticides. Things like that. So we're really trying to develop a very sustainable system. Rocco mentioned this NCAP program, which is a free program through NREL for commercialization for small businesses. And so what they've done is looked at the production and purification of our cyanobacterial sugars. So just starting with that and building an Excel model to estimate the key mass and energy balances. They quantify all the associated capital equipment and operating costs for that equipment and to operate the farm. And in the end, come up with a minimum sugar selling price, which would be the benchmark compared to, say, corn and cane or the cellulosic sources. Some of the features in the model are the biomass propagation and production ponds is one unit of the process that we would need to do the economics involved. And then there's the separation, the centrifugation, removing the biomass from the salty sugar water. And then the sugar recovery and purification process. That's what Rocco was talking about with using some sort of microfiltration or reverse osmosis or some other technology. And again, where the engineering really needs to come in and develop and optimize a system that's going to allow you to economically separate those sugars out and get those into a fermentation process. And then also looking at the co-products and waste products. And they looked at, what would be our waste products and what other products coming out of the system could actually have economic value versus being an economic deficit. And I think in any one of these biofuel or in this entire segment, trying to replace oil, you really have to eke every dollar out of the system you can. You have to be as mass and energy efficient as you can to be able to compete with oil because they have that 100-year head start. They've got embedded subsidies and tax incentives and things like that. So to make this work, and a lot of work that went into our model is based on, NREL has done a lot of work with algae and biodiesel. And so our system is not dissimilar in that you've got these open ponds and you've got some of the similar, water processing, water handling, things like that. It's all a very similar kind of system. And what they've discovered is that the economics do not work unless you can use these co-products and develop what we call an integrated biorefinery. And that's sort of an industry standard right now of how people are trying to put all of this together, that it's not just one crop, one product, but it really has to be this, ecomimicry based concept where you're putting all of those elements together to make the economics work and to make it sustainable. So somebody asked, one of the questions was about, one of the questions that was submitted were about what are all the different aspects of this process and what are the carbon costs of this and what's the sort of the energy return on investment. So Life Cycle Associates, sort of, some people that we know from locally put together this greenhouse gas analysis. So looking at the greenhouse gas emissions of the different steps of our process compared to petroleum gasoline. So chemicals and fertilizers, like I said, almost nothing. We're not using those at all. Very, very little bit of trace elements that we might need to supplement the seawater. The cultivation, the pond systems, there's some greenhouse gas emissions associated with pumping the fluids around. The separation desalination turns out to be a big cost. There's a lot of energy that's required to run those processes. And that's where some significant gains could be made, both in terms of greenhouse gas emission reductions, as well as in terms of the economics. And then the biofuel production, the fermentation systems and things like that have to be incorporated in if we're going to look at all the way to gasoline. And this is something that we wouldn't have direct control over, but that would be our partners. And then biofuel transport and delivery. One of the things that we've talked, built into our model is that we want to be co-located with our partners. You can't ship seawater, into the middle of the country, and you can't ship dilute, sugar solutions around. So you really want to have that fermentation system right there. And there's other advantages of that too, because off the fermentation, you can take the carbon dioxide, run that back into your propagation system, so you're not wasting the CO2 coming off fermentation, but you're actually using it as an input to another part of the process. And then, of course, there's greenhouse gas emissions with the actual combustion of the ethanol. So we compare very favorably in this analysis with gasoline. It would give us an advanced EPA biofuel rating, which would get us some of those RENs and those other credits and things like that as a process. Question?
Yeah, so you're saying the greenhouse gas emissions from the energy needed to process this, what kind of energy bases are you using? Is this for like a coal-based or?
So everything, all this section is for cyanobacterial sugars fermented by yeast to ethanol in a traditional biofuel plant versus petroleum gasoline versus burning just gasoline in a car. Right, so you're comparing, the ethanol that you would use if you had a car that was, around E100 car, or just using that, just the energy embedded in that ethanol. How much energy went in and how much greenhouse gases went in to getting that same amount of fuel that you would from a gallon of gasoline.
Is that not the energy of processing this? Is the CO2 that it would emit when you use
it's the total greenhouse gas emissions of the whole process. So if you're pumping fluids, it's the electrical energy to pump the fluids around. If you're using plastic pond liner for your propagation system, it's the embedded energy in that pond liner. So it's sort of trying to capture the cradle to grave greenhouse gas use of our process compared to gasoline. Does that answer your question?
Yeah, no, I was just considering where you have your plant, then the energy, like the CO2 by energy they use is going to be different. So I guess
have they burned coal or have they burned oil or is it solar?
Oh, I see. In terms of where we're getting that electrical energy, right? Yeah, but that's not in here. This model is not at that level of detail.
No, it isn't. In fact, if you look up there, you can see that it's actually energy. Part of the energy conversion is in megajoules. So it's not, and it's not based specifically on coal. It's not based on or anything like that. It's more generic and it's not that detailed. Does that make sense? You know what I mean?
So it will change depending where they locate.
Yeah,
right. For example, if you're able to use your biomass in an anaerobic digester and generate your electricity locally, right, then that's going to have significant advantages versus if you're using, dirty coal from China to run your process. No, that's a very good question, and we are not at that level of detail in our this analysis right now. This was kind of a free exchange of ideas from these models are pretty well developed, but until we would have more of a system with the specific details on that facility, we wouldn't even have the information to put into a more detailed model. So just briefly wanted to give you an idea, it's not just all, schematic diagrams, that this is actually, we're actually making real progress towards getting this done outside. So Moss Landing Marine Lab, as Rocco mentioned, is down near the power plant in Moss Landing on your way down to Monterey. And right out here, they've built a new aquaculture facility, which has access to seawater, and we would, have a couple thousand-gallon tanks that we could use to put our consortium of cyanobacteria into those tanks, measure those. We can then take we could measure our productivity, we could look at the scale-up issues, how it works, at larger volumes, and then do all of the sugar analysis and microbiology analysis with SRI and LBL support. Now, Monterey Blue Water Farms actually now has leased this facility, which is right next door to the power plant, has existing ponds, has an raceway ponds here, has some smaller ponds here. We would partner with them and be able to use those facilities to bring in see what they have also, the permits and all the regulatory issues have been resolved to get seawater into here and also to dispose of the seawater out of there. So we could use these existing facilities. So we actually would be able to bootstrap ourselves pretty quickly to going from a few thousands of gallons up to a 1 acre pond. And our strategy is to have a 1 acre pond as our sort of our base unit of operations. So get to build and operate and manage and characterize a 1 acre pond system. And then much like solar panels, when you want to scale up, when you need larger volumes, when you want to get to the Chevron scale ability to meet that demand, like solar panels, you add more panels, right? You don't make bigger and bigger ponds. You just add a modular pond system that is well characterized, well understood, because the pond management is really important, and you need to have the growth, you need to have the reliability of those systems. So our strategy is to bootstrap off of some existing facilities and abilities down there, and then once that's proved out, would be to attract a commercial partner to work with us and to do an offtake agreement, and we would co-locate with them. This happens to be a pond farm down in Baja California that we just plucked off Google Earth, and tens and hundreds of thousands of acres of desert that could potentially be suitable to get to the kind of scales that you'd need to be able to produce enough, fuels to initially get a niche market into the bioproducts market, to get into biofuels. For example, we scaled it out. The plant up in Richmond is 50 million gallons of ethanol per year is what they're mandated by the state to use. That would be a 5,000 acre facility. Probably not very likely we're going to get a 5,000 acre facility that's cost effective in Northern California. But in Baja California, coastal China, in the Arabian Peninsula, North Africa, places like that, you would be able to build facilities of a sufficient scale without the land, freshwater, food competition issues that would start to be able to at least make one of those stabilization wedges of addressing climate change. I don't know if that's a concept that you've talked about in your class, but basically the idea is it's all got to work, right? To really keep the oil in the ground, you've got to have solar energy that works, energy efficiency and conservation. You've got to have some biofuels. You've got to have oil replacements. Somebody talked about electric cars versus transportation fuels. I think you need all of that to work, and that's sort of where we're headed. Yeah, please go ahead.
So a place like Baja would be a pretty obviously good location, but a place like Maine, someplace with a hard winter, probably not so good?
Probably not, yeah. Probably more, equatorial, or somewhere within that band. And in places, and you can think of it as also opportunities for rural economic development, having a facility that is, and that the last slide after this when we get up there, just really quickly, Rocco talked about the desalination. A lot of different options we've looked at, a lot of companies that we've looked at that are putting these things together, looking at both the energy costs, which tends to be about 6 cents a pound out of our projected maybe 12 to 18 cents a pound, and about 4 cents a pound for the capital cost for that equipment. So it's a big bite in the economic model, and that's an area where engineering and biology need to work together to be able to solve some of those problems and come up with solutions that are going to make that work.
Salt separation type of work?
No.
Yeah. We should talk about that.
Okay, yeah, I would love to, know what, other options might be. And last slide, I know we're probably coming up against our time limit here, but this is our concept of the integrated coastal biorefinery. This is sort of the big ecomimicry concept where you're growing your cyanobacteria in these ponds, you're separating it, you're taking your salty seawater, you're processing it through some efficient process, and you're getting this feedstock that can go into algal growth, can go directly to bioplastics, it can go into fermentation and distillation process to produce an ethanol, chemicals, and things like that. That CO2 feeds back into your ponds. The nexus with the aquaculture industry is that there's a lot of nitrate and ammonium that comes off of their fish farming, and before they can dump that back in the ocean, they've got to get rid of that. Our cyanobacteria could eat up that nitrogen waste and put fresh, clean up that water for seawater return, amplifying the biomass, and that biomass coming from both, all these different sources, then becomes an input for the fish food. So a very nice, tightly coupled mass and energy balance system. If that biomass isn't used for fish feed, it could go into anaerobic digestion, for example, is one option that we've looked at that would be able to produce energy, and you can get some reasonably good compost out of some of the wet anaerobic systems that are commercially available. So looking at building a system like that is way beyond anything I looked at NASA for a Mars mission or something like that. It's really a complicated biological, ecological, and engineering problem, and it's great to see people interested in pursuing trying to solve some of those problems, because a couple of microbiologists on our own, we're not going to do it by ourselves. We're going to need a lot of support. So I'm happy to open it up for questions, and however you want to proceed. So thanks.
I just always wondered, so do these ponds, do you keep them as a batch reactor, or do you run it constantly in and out?
No, the idea would be a batch operation, and we're looking at about maybe a 3-week residence time. So, and that's another reason to have a lot of smaller ponds, because I can tell you from just doing laboratory work that you can do things exactly the same with bacterial cultures, and sometimes it just dies and it just doesn't work. And so you're going to have you're going to have some subset of those ponds that are probably going to be in the process of being cleaned out, or the culture crashes or gets contaminated. So you want to have redundancy built into the system and these batch processes so that you're being able to provide a constant, steady supply of sugar to your partner there who's doing fermentation or processing to whatever end product. Yeah.
So last summer I peer-reviewed a paper on like a study of animal interactions with offshore ponds. Essentially, it was like a box they left out, and they were like testing just to see like how seals, how any other kind of animal, seagulls interacted with it. And that was a pretty boring study in my opinion. I mean, it didn't go over a lot of the other possible pros and cons of it. And so I was wondering like what is preventing you guys from doing offshore? Like, just the pros and cons in your opinion.
Well, I mean, I think trying to manage. So there's a guy actually who did his work at Ames that was talking about floating bags in the ocean, where he would put have these floating bags of algae out in the ocean, and it would be thermally regulated, and you'd be able to get your sun from that, and it's a complicated environment to work in. It's a really, really rough, rugged environment. I dabble a little bit in surfing. I can tell you it could be really, really damaging environment. So it was never a consideration of ours to do offshore, aquaculture type.
It obviously wouldn't integrate well if it was offshore.
Right, yeah, no, it wouldn't integrate well. And the idea, the near coastal areas, I think could work really well. But to try to grow it out in the ocean and manage it is, I just think it would be cost competitive.
Impractical. That project they spent, the Omega project is what it was called. They spent several $1 million before they finally realized that it just was too impractical and not going to work. And the growth of the organisms wasn't all that efficient either. They had problems with the bags. They had problems with growing the organisms and efficiently, photosynthetic efficiency, and all. They had organisms shading each other. It was messy.
Yeah. And there's been a lot of research done, for probably about 35 years on open ponds outdoors. One of our ideas, though, is that probably to put some sort of covering over the ponds, like you see a lot of the greenhouses. It's just really, PVC pipe with plastic cloth over it to be able to keep a lot of stuff from getting into the pond, so you don't have problems with contamination as much. Keeping out predators, because sugar is a real commodity out there in the wild world, and so anything that gets into your pond that eats that up, which is why we want to keep the residence time short, is so that we get in there, we find out what the optimal time is when the sugars are at their peak, and then get them out of there, reset the pond, and start over. And that's where the real empirical work outside in the lab needs to prove that this can work, and then I think that would where the more outside investment would come. Another question?
Thinking about like covering it, would that affect, because you need the light to come in, right?
It wouldn't be, it's more like a screen.
Yeah, it could be a mesh, right?
It is a mesh, and we've done this.
Oh, I see.
And actually the organisms grow quite well. Because they are so photosynthetically efficient, they don't need quite as many photons. There is a problem sometimes where you can get them to get too many photons, and there's photobleaching. So it eliminates that. The other thing that the covering does not only keeps out a lot of things, but it also decreases the evaporation. Evaporation tends to be a problem.
What about selective, like solar cells? Is there a certain wavelength range that the cyanobacteria uses?
Yeah, 400 to 700 nanometers.
And so, theoretically, would it be possible to have like a selective solar cell, like transparent one that is?
Oh, you mean sort of couple electrical energy generation with
absorbing a range.
Sure, that would be a great idea if you could have sort of a porous mesh over that would be the physical barrier you need, would let the right wavelengths through, and then capture the other parts of the electromagnetic spectrum and use it for electricity. That would be a great add-on to that.
Are you guys familiar with building 239 in NASA Ames?
That's where we worked.
Yeah,
Very familiar.
Yeah, on the fourth floor, I think they were experimenting with that, or just on the rooftop. They had
and now right in front of the building or near the parking lot, they have a solar panel that's about as big as that wall, or almost as big as that wall.
Yeah, I spent a lot of summers working there. So
Yeah. Okay.
on the fourth floor.
Okay. Good. No, that's it. Good idea. Yes.
What do you plan to do with the brine that's generated in the feedstock production?
That's a really good question. If you could help us solve that. That's a very good, I mean, one thing that they've talked about down in Monterey, the aquaculture guys, is that, is to bleed their return water back down in deep and slowly so that it doesn't cause localized salination and disruption, because that's a very sensitive marine ecosystem down there. So the California Coastal Commission in Monterey County, the regulations are very tight. But, accumulating brine over time, it's really a challenge with what to do that. I mean, some of it could go maybe into industrial salts and things like that, but it's an area that we have not put a lot of effort into yet. It's just, there's a lot of moving pieces here, but that's a good catch, because you can see that there's no arrow coming out. Exactly, yes.
So how is this different, like taking seawater and desalinating it instead of just taking normal water? Like, why do you?
Well, because the sugar and the salt molecules are very similar in molecular weight and size. So that's one of the complicating features. You need to design a membrane and a process where you get the salt on one side and the sugar on the other side. Now, if we could get pure water, out of the desalination process as another element of the integration of that, obviously fresh water is a commodity, and especially in desert climates, that could be a real commodity. It's something that we've thought about, but we haven't, had taken as deep a dive on that particular question as we just haven't had the capacity to look at that. But I think if fresh water was another product that came out of all of this process, because you're talking about millions of gallons of seawater in a large facility, and if you could also produce fresh water and use that for local agriculture, local drinking water, that would be good, and you would still have the salt accumulation problem, and that would be something that I think would need to be looked at, because I know it's a problem for a lot of desalination plants is the salt accumulation. But then again, Cargill spends all this time and effort in the South Bay getting the salt out of seawater and selling it. So maybe we become a competitor with Cargill for industrial salt.
When you go through this process of separating everything, like what happens to your cyanobacteria? Does it get discarded or do you reuse it?
So that's the biomass. So that could become fish food, or that biomass could go into anaerobic digestion. There's some open questions about the suitability of cyanobacteria for the different kinds of fish that they're growing. But out in the open ocean, it's a food source for a lot of things. So our assumption is that there's going to be a way to use that biomass to feed the fish in this operation. If we didn't have the aquaculture, then our original plan was to take that biomass through anaerobic digestion and create electrical energy and compost.
Well, this is great. Thank you so much.
Well, you're very welcome, and great questions, and it's nice to be able to,
Let's thank her.